Um, thank you very much. So
Yes.
I'm delighted to be a part of this discussion. George made my life easier by talking about dengue, and he also said about the antibody dependent enhancement problem of dengue vaccines. We call them villain antibodies. So we all know that vaccines are the most cost effective public health tools. Maurice Hillman, he's the father of vaccinology in my view. He made 34 vaccines in his lifetime. So making a vaccine was not very hard. You take a pathogen, weaken it, and you have a vaccine. We are all alive today because of Maurice Hillman, because almost all of us have taken all the vaccines in expanded program of immunization. All the 8 vaccines which saved billions of lives were made by him. But dengue has been a very difficult story. Dengue vaccine have been pursued and supported over decades by WHO, by NIH, by several organizations. And even after about 20 to 25 years of research and clinical development, when Sanofi launched dengue vaccine, it created hell. This is the picture of the parents who are carrying the photographs of the kids who seems to have died because of the bad antibodies which were produced after dengue vaccination. So my talk is that why, after so many years of support and research, a safe dengue vaccines have remained elusive. While we have seen within a year, a COVID vaccine became such a great story. Of course, it is the cooperation, the international cooperation and the funding and the political will came into this. And we do know that the messenger RNA technology may be the future. And India, according to WHO, is the biggest, has the biggest production capacity for vaccines, and it is the biggest asset that world has. And we believe that next to US, for example, if we take example of COVID, in 2021, 4 billion COVID vaccine will be made in US, while 3.5 billion vaccine for COVID will be made in India. The mRNA vaccine is also being produced in India. It is going through now phase 2 trials. And the good thing about that vaccine is that it is stable at refrigerator temperatures. So let me come back to why safe dengue vaccines remain elusive. And the bottom line is that dengue antibodies can be heroes, which protect you from the disease, or villains, which cause the disease, which enhance the disease. That's what George mentioned. So it's a mosquito-borne viral infection which can cause dengue fever, dengue hemorrhagic fever, and dengue shock syndrome. So in this overview, I'm going to cover 3 key topics. One, what is dengue disease and dengue virus? Why safe dengue vaccines have remained elusive? And my bottom line is because all these vaccines produce bad antibodies, and we call them villain antibodies, which help the virus to infect the cells to a higher load. And how a designed dengue vaccine candidate can teach immune system to elicit only hero antibodies, which will block the virus infection in complete absence of villain antibodies. So dengue still remains a serious public health threat. And this picture shows that you could have, just sudden onset of fever with headache. This could be mild dengue. You could have a mouth, nose bleeding, muscle pain, vomiting. You could then go through some critical phase. You could have low blood pressures. You could have GI bleeding, and then you recover. But somehow, because of the presence of villain antibodies, the dengue fever can turn into dengue hemorrhagic fever, or it could turn into a dengue shock syndrome, and you could lose a patient within a day. So dengue virus is a single-stranded RNA virus, and it has about 3 structural and 7 non-structural proteins. There are 4 serotypes. That's a major problem with dengue. These, if there was one dengue virus, we would have had the vaccine without any problem. But since there are 4 serotypes, and these serotypes, the antibodies to these serotypes seems to cross-react, but do not cross-protect. They seems to cross-enhance the infection. So each virus can cause dengue fever, dengue hemorrhagic fever, dengue shock syndrome, lifetime immunity to a single serotype, secondary dengue infections could be serious. So we need tetravalent vaccines. If we look at the structure of the virus, this is the genome, and it makes 7 non-structural proteins and 3 structural proteins. And from vaccine point of view, the preem and the envelope are the main target, but immune responses to dengue virus infections are dominated by villain antibodies. About 80 to 90% antibodies are made against the membrane proteins and the fusion loop proteins, and the hero antibodies, which are very low in a natural infection, are the one which can protect the host from infection. So complexities of dengue viruses have been that, of course, there are 4 serotypes. If that was not enough, each serotype, because it's an RNA virus, there are various genotypes between each serotype. And to top it off, we now know that the virus seems to breathe in your body. That is to say that when it comes out of the cells, infected cells, it has various forms. It's fully infectious virus, which can infect the cell through its receptor binding domain. Then they are partially infectious or non-infectious viruses. They cannot infect the cell in absence of an antibody. This is where the bad antibodies, the villain antibodies, can help these non-infectious viruses to become infectious, and you could have a severe disease. The best thing would be to avoid these villain antibodies. Now, how do villain antibodies help non-infectious virus to become infectious? George did mention partly this, that normally what happens is that the dengue virus can enter through its receptor, and then innate signaling is on, and you have type 1 interferon pro-inflammatory cytokines which kind of control the virus replication, and you have in this side of the system the virus is kind of controlled. But what the virus which comes out has now different forms. Only the fully infectious virus can come back here, but because of the interferons, the replication is stopped. But it is the other kind, non-infectious or partially infectious viruses come through the FC receptor, and this is what I'm calling them villain antibodies, and they suppress the cytokine signaling, and now the virus is free to replicate to much higher level. The production of virus from this side could be 10 times higher, and this is why the secondary infections are very severe, and these are villain antibodies cause immune evasion by dengue viruses. So villain antibodies, in our view, cause the vaccine failure in dengue. All the whole virus-based vaccines use 4 viruses, and they elicit a huge amount of predominantly villain antibodies, which are shown here by dashed antibodies, while the hero antibodies, which are solid antibodies, and 4 different colors are being shown that they are serotype specific for dengue 1, 2, or 3, or 4. But the cross-reactive pre-M and fusion loop antibodies are a major problem of vaccine failure. How do we avoid vaccine failure? We believe that if we avoid these villain antibodies and we just produce hero antibodies, and so for producing hero antibodies, we use only the receptor binding domain from our design, and we remove the pre-M and fusion loop epitopes. So these are designed vaccines, which will only produce serotype specific antibodies, which are hero antibodies to dengue 1, 2, and 3, and 4, in absence of any villain antibodies. So how do we design such a vaccine, which will only produce hero antibodies? Here is a cartoon of the whole dengue virus. If you look at the blue domain is the receptor binding domain. If you chop that domain off by trypsinization, the virus will stand outside the cell. It will not be able to infect the cell in absence of an antibody. So we use RBD of each of the 4 dengue virus serotypes, and we link them together. So instead of using the whole viruses, which everybody else uses, we use the business end of the virus, which is the receptor binding domain, and through genetic engineering, we link them into a one protein, and then we link it with the hepatitis B surface antigen, produce them in Pichia-Pastores to high quantities, and these virus-like, single chimeric virus-like particle is able to make antibodies to hepatitis B surface antigen, to dengue 1, 2, 3, and 4. And so this DSV4 vaccine teaches the immune system only to make hero antibodies. And this is the design of the vaccine, that we have the receptor binding domain from 1, 3, 4, and 2, all the 4 viruses linked with surface antigen. The VLPs are produced, and when we have checked them, and they produce serotype specific antibodies to each one of them, and we call them hero antibodies. If we remove dengue 4 from our design, there would not be any neutralization of dengue 4. And similarly, if we remove dengue 2, there won't be any neutralization of dengue 2. This cannot be said for Sanofi. If you depleted dengue 4 antibodies, all the neutralizations will disappear, which meant that the whole virus-based vaccines are neutralizing viruses by cross-reactive villain antibodies versus the hero antibodies. How do we score? Is there a way to check for hero or villain antibodies? If we do the neutralization only in a test tube or in a tissue culture, both the hero as well as the villain antibodies will neutralize the virus. You will not see anything on the in vivo cells. But in vivo, antibody-dependent enhancement is an in vivo phenomenon. It's not an in vitro phenomenon. So for that, there is a mouse model system in which one can add a sublethal dose of dengue virus. The mouse survives. This amount of virus is not lethal. But if you mix it with the villain antibodies, now the virus is completely neutralized. So here is a completely neutralized virus with the fusion loop neutralizing antibody. Now, no plaques on the vero cells, but the mouse is dead. This is what we call antibody-dependent enhancement. Apart from that, you can see fever, thrombocytopenia, hemoconcentration, vascular leak, cytokine storm, increased virus load, means all the symptoms, clinical symptoms of dengue you can see in this mouse model. But if you neutralize now with the receptor binding domain, which we call hero antibodies, or even if you partially neutralize, the mouse survives. So subpathogenic dose can be converted into pathogenic by villain antibodies, but not by the hero antibodies. Here is a video which we recently published in eBiomedicine this year, wherein we had a head-on comparison of Dengvaxia, which is a kind of an approved vaccine with a limited approval. We did not have access to NIH vaccine or Takeda vaccine, but we made our in-house tetravalent dengue vaccine by mixing 4 viruses, live viruses. So that was a surrogate, a lab version of the Takeda and NIH vaccine. And of course, our ICGB vaccine we call DSV4, which is dengue subunit vaccine tetravalent. And when we take these seras and we make 100% neutralized immune complexes of the dengue virus, put them on the vero cells, each vaccine shows complete neutralization. But once we put them again into the mice, you would see that The mice are healthy when they're injected with the immune complexes made with the ICGB vaccine, but neutralized immune complexes by Dengvaxia antibody or by the whole tetravalent virus, the mouse are the mice are showing enhancement. So these are the villain antibodies. Despite in vitro neutralization, there is an in vivo enhancement. So this is where the problem is, that if we just check them for their neutralization titers and we say, okay, they neutralize, but we do not check them for in vivo enhancement, we will end up into the same story as what Dengvaxia has done. We have seen this thing happening with the secondary human sera. If we bring the patient sera, it also causes enhancement despite causing neutralization. So why do these mice kind of show enhancement and die if you cut them open? And this is what was happening in Philippines. Those kids who were dying when the surgeons were doing the postmortem, they saw their intestines were bleeding. And here we see in these mice, when they were injected with the ICGB vaccine candidate, when they only produced hero antibodies, the intestine looked very nice. But with the villain antibodies produced by Dengvaxia or the whole virus-based vaccines, they were bleeding inside. And because of that internal bleeding, the mice were dying. So this is my last slide, more or less, that our aim is to develop a safe, effective, affordable, genetically engineered dengue vaccine for use in all age groups in dengue endemic and non-endemic areas. This is what happens, I'm showing you by a cartoon, natural infection, or I believe with the whole virus-based vaccines, you have very small amount of hero antibodies, which are good, but dominated by villain antibodies, which are bad. And ICGB vaccine candidates turns this table around, and you only have hero antibodies in absence of villain antibodies. These are my collaborators across the world. I have worked with Global Vaccines, with Emory, with UNC, with Johns Hopkins, and we have done some work at Mount Sinai. Because dengue and Zika cross-react, our vaccine does not enhance Zika, unlike the Dengvaxia or the tetravalent whole virus-based dengue vaccines. And the work has been now going on with Sun Pharma. We got a lot of funding from DBT, from Wellcome Trust, from NIH, and from BIRAC, Government of India. Nothing of this was possible without having some of the best students with ICGB provides us, from within India and from various other countries. And thank you for your time and attention.
Thank you very much, Dr. Khanna. Certainly a disease that we don't know in this part of the world, but it looks like the immunology and the research is important, and we will probably see more of it. So it'll be interesting to see the technologies and the products that come from this. There's just a quick question here. How do you, using antibody tests, can you actually distinguish between the hero and the villain?
Absolutely. We cannot distinguish them on viro cells. You can, both hero and villain antibodies, they neutralize the virus. And if you put this neutralized viruses in the viro cells, you do not see any plaques. But once you put this neutralized immune complexes into this AG129 mouse model, if the mouse survives, they are hero antibodies. There is no vascular leak inside. If the mouse dies, they are villain antibodies.
Wow. Okay. Yeah. All right. Luis, do you want to say something?
No, it's fine.
Okay. All right. That's fine. Thank you very much.